Our first speaker for today is Leo. Leo is from Estonia and transferred after 2 years at University of Tartu to finish his studies in mathematics as well as computer science and engineering at MIT. He then did his PhD in the UK at the University of Cambridge and the Wellcome Trust Sanger Institute under Richard Durbin, and has been working with Brenda Andrews and Charlie Boone since then. His PhD work was in devising new statistical experimental methods for genetic mapping, and he's currently working on the same for protein traits in yeast. So, I will turn it over.
Hello. So I'm Leo, I work here on the 13th floor, and I care mostly about mapping heritable traits. And while this thing starts up, so it's sort of obvious that most traits are heritable, or what it means is that related people tend to look more similar in a trait than unrelated people, or children look like their parents. And the genome can sort of manifest this heritable trait through mRNA levels and then proteins. So mRNA level and message. So the only two ways the genome has an effect. And so we all know also for this most basic traits how heritable they are. So mRNA levels in yeast, more than 50% of the genes have mRNA levels that are at least 50% heritable. So it's quite strongly heritable. In human, the estimates right now are around 20 to 40% in different tissues. But there are a lot of things that we don't know. After mRNAs, which is sort of a slightly uninteresting molecule, it decays rapidly. There's maybe, again, in yeast, most mRNAs are present at less, okay, let me be more precise. More than 80% of the mRNAs are present at less than 10 copies per cell on average, whereas proteins are present at tens of thousands of copies and actually do all the function. But we don't really know much about the protein levels, protein localizations, and also metabolite levels and metabolite fluxes, because we can't measure them so easily. So, how to understand the heritability of traits? I think the protein traits are the most interesting ones to study because they're this sort of tangible intermediate between the broad traits that we care about and sort of the basic mRNA traits that are already well studied and but maybe not as interesting. So the way to do this is we use natural variation, so we use naturally occurring alleles in the big wild population in yeast. We do large-scale yeast crosses using Charles SJ technology. We do protein measurements in high throughput using microscopy and flow cytometry. And finally, we do bulk genotyping of these pools. So, how do you measure protein levels in yeast? You can pretty much only do it in yeast properly, at least in single cells. And the trick is to just visualize it. So you can't get a nucleic acid readout, which you could sequence. So the other way around is to just look at it in a microscope. So in yeast, how it works is if this is a big yeast cell, this is the yeast genome, this is your favorite yeast gene, you can attach a cassette that has the green fluorescent protein in it and then some selectable marker to the end of a gene. So if you have this yeast cell now and you have it growing, then it will produce this protein, and the protein can be excited and visualized under a microscope. For me to apply these linear models that are amazing and cool, but also simple enough, so I need these populations so I can measure and I can fit these models. So the way I create these populations is take a father, that's a wild strain, that's this natural genetic variation bit. I take the mother of the fluorescent tag and I cross them. So the kids, I'll have a very large population of them. They will look something like this. They'll be spread in the GFP values because it's a heritable trait, the protein level, and their genomes will be a mix of the two parents. So if say red is the color of the mother and blue is the color of the father, if you look from left to right the entire yeast genome, you'll get blocks that come from mother or the father. So it's a mixture. As a control at the same time, we take the mother, do a sex change, cross again, and get this sort of control population. So this is what we use to estimate the sort of clonal variability.
Where does the wild strain come from?
It comes from a California vineyard. It's a well-studied strain from like the early 2000s, and there's a lot of previous data on it. That's why I use it. So if you compare the kids from the two populations, then this control population will have all looking like the mother, like all genome will be red. All right, so we have these for one protein. So that was experiment for one protein. We repeat this for every protein that we have this cassette for. So that's almost 4,000 proteins in total, and so that's where you need the Charles SJ technology, large scale crossing and so on. So this is what it looks like under a microscope. You'll have cells. The red here is a nuclear tag, and you see that some cells have more, some cells have less GFP. You have to show them me when I'm going over time. I have like 100 slides. So now the bioinformatics aspect of all this, right? So we can do the screen, we can take the images. There's a lot of images, they fill terabytes. It's horrible if it gets lost and it takes 2 months to restore. So the informatics problems here. You have these images, you have many of them. There's a lot of biases between images. So it's not straightforward to go from images to actually understanding what the cells are. And you obviously have to do it in an automated way. You can't sort of trace outlines around these things or sort of manually quantify how much GFP there is. So one has to find a good way to correct for any batch effects between image and plates, to differentiate between background and foreground, to segment the cells out of the background. And that's a good bit of bioinformatics work. And what comes out is about 50 to 100 cells per image, and for every one of these protein strains we get about we do exactly 4 images. So in total we get to 200 to 400 cells out. So, and this is what it looks like. We remove the background, we've done the segmentation. So both for nucleus and the cell outline is not the same picture. And So what we do then is we go from these cell pictures and these cell objects to a large sort of feature matrix. So here, every row is one cell, every column is one feature. We have, again, 400 or 500 features and hundreds of thousands of cells, and we get this huge feature matrix. And so what we do then, obviously, is to create a training set. So for every protein localization, like this one seems to be membranes, we have about 20 of these classes: membrane, nucleus within the cell, small dots in other places, some more small dots. They're mostly small dots. And yeah, and here you have all these machine learning problems. So how do you deal with this very high dimensional feature space? Do you need all the features? Do you need to do feature selection? Can you do dimensionality reduction first? So yeah, so there is a lot of sort of problems to play with at almost every step in this process. And that's very much work in progress. What we have made a lot more headway on is using flow cytometry as a different kind of readout of the GFP level. So you don't get the localization info, you don't get a picture, but what you get is a quantitative measure of the protein level. And the way it works is we take the same cell population and we put it into this into a sheath fluid and through a nozzle past the laser. And the laser excites the GFP, and it also is deflected by just the cell itself, and we get out measurements about the cell size and GFP level. So it's a lower dimensional readout. We get readouts from about 16 channels, and we don't use all of them. But the upside is that we can measure a lot of cells. So this is what that looks like. So this is a measure of cell size and the cell complexity on the x and y axis, and each dot here is one cell. So we get something like 50 to 100,000 cells per experiment. And we can filter the cells that we deem are good, like all those guys are sort of too broad and too wide or too complex. So we can distinguish between our control experiment and the wild strain cross here. So those are those two populations. And then we can actually finally get to our readout. So this is now the GFP level on the x-axis, and just the frequency on the y-axis. So we wanted this. This is what we got. This red here is not actually the real color. But the point is that the parents look like this and the kids look like this. So basically the parents and kids really are different. So this protein level is heritable. So again, we have these now for 4,000 proteins. And most of the time this looks sort of like this. If you do this quantification, you see that those bumps are pretty much straight on top of each other. So we can condense all that data, 100,000 cells, different filters, and the GFP measurements onto a single number. So what's the median GFP level in some fairly well, cleanly defined population? And we get this number for the mother, and we get this number for the kids. So the natural question is, how different are the kids from the mother? And the answer is not very. So if this is the diagonal here, you can hopefully still see that these somehow faded dots, there's not a lot of them that are off diagonal. So most protein levels in this cross, where the mother and the father are about 1% different in their genome, most protein levels are not that different, so not that heritable. But there's still plenty of things that are off diagonal. And so these measurements are reproducible if these are two technical replicates of the difference in the x and y axis. So difference between mother and kids, difference between mother and kids, but different replicas. Even small difference, so something like 0.1, which is a change of 25% in the absolute protein level. So these are fairly well replicated. And based on this, we estimate that there is roughly 250 proteins that are sort of strongly heritable in this cross out of the 4,000 that we can measure. And we can turn this difference into an actual value of heritability. So what fraction of total variation is due to heritability? And so this is sort of the history on that. How much time do I have? Not a lot. Negative minutes by far. We do a lot of cool stuff, basically. And this is the most important slide. It's been a good collaboration with Adam Rosebrock, and I work in Brendan Charlie's labs, who provide tools and other things. And Amy and Candace have helped out with creating strains and so on. Joe and Fred's with sequencing. Dion is sorting. People in Charlie and Brendan's labs with answering my endless questions, and CFAR for funding. Thank you very much.
Are there any rapid questions for Leo as he goes through his?
Yes.
How do you know that GFP tags aren't being lost as the children have children?
Two ways. Firstly, we can see the GFP. Like if there's no GFP, you can't see the protein. And secondly, they're also constantly under selection. So we select for the bit that comes after the tag. So they have to be able to survive without histidine. So.
Any other questions for Leo?
Good, all clear.